the ACER projects that have been looking at the impacts of Deepwater Horizon oil spill over the last 10 years and trying to synthesize them into what we learned. And so first what I'm going to walk you through today is what happened during the oil spill, then what really ACER focused on, and give you some highlights about one of the groups and what their research did, and then some brief findings of what I've been finding in my synthesis. Okay, so 10 years ago, the Deepwater Horizon well blew out, and it released oil for over 80 days and until it was capped and then eventually sealed. And so the first responders worked really hard to try and minimize the impacts of these oiling by essentially trying to corral the oil and remove it from the surface. And so they did that in a whole host of different ways. So we have our oil slick. As you can see, oil floats. And some of the methods that they would use are physical structures like booms, where they would try and corral the oil. But as you can see, some of it can kind of go over top of it, especially if there are waves. They also used different things on the surface to try and absorb the oil. So you can see as my little absorbent strip gets more brown, I'm picking up more of the oil. And lastly, one of the other things, well, they did also try and burn off oil on the surface, but I'm not going to try and do that for you today. One of the other things they did do is add in dispersant, so soap in our case, and as you can see, it just makes the oil move and becomes more like smaller clumps. So it's kind of breaking, oops, everything's flying, breaking down those particles until smaller and smaller oil drops. And so this is some of the oil that was from the spill, and it's been in this jar for 10 years. It's weathered, which that means basically that there's both been photooxidation on this oil and also it's been physically moved. So as you can see, it's more like clumps, less like slicks that we started with in the tub.
And when you say oxidation, photooxidation, what do you mean?
Just that the light breaks it down. It's a chemical reaction. And so also what other things, so although all these people did all these different types of tasks, the booms, absorbent, dispersant, we did have oil that, whoops, sorry, I'm going to take off my gloves now. That might be helpful. So we did have oil that came across the shoreline. So approximately about 2,000 kilometers of shoreline in the Gulf got different amounts of oil. So the red in that picture is pretty much heavy oiled sites, and the lighter colors become less and less oiled. And so some of the oil that made it to shore came in the form of these tar balls. And essentially what happened after this is that scientists from all around the Gulf and their collaborators wanted to look at what were the ecological effects of this oil on these different coastal and deep water and pelagic habitats where this oil spill actually occurred. And what they're really focusing on is trying to see if these habitats, if they would go back to their natural or not natural, if they would bounce back and go back to the way they were prior to the oil spill. So we call that resilience. The ability of this ecosystem or area to bounce back from a disturbance. And we can kind of think about it with this ball and cup diagram. So if the ball is where you started, if oil or any kind of disturbance, so disturbance can be both natural, like a hurricane, or man-made, like an oil spill, and the disturbance pushes the ball. If it's resilient, once it goes up, rolls up the hill, it'll roll back down after some amount of time, so it will recover back to that initial state. But sometimes what happens is the ecosystem gets pushed up over the hill and it'll settle somewhere else into some different ecological state. And so the researchers here at Dolphin Island Sea Lab were part of this consortium called ACER, which is the Alabama Center of Ecological Resilience. And what we were really interested in focusing on was, does diversity help in this resistance of ecosystems and resilience and recovery to this oil disturbance? So first, I'll add why we were interested in looking at that, but first I'm going to talk about what is biodiversity so we're all on kind of the same page. Okay, so if I first asked you what is a diverse ecosystem, probably the first thing people think about is either tropical rainforest or coral reefs. And what you're really thinking about is these places or ecosystems that have high number of species. So either in this case, a number of coral species, which might be able to see in the picture, or a whole bunch of different fish. And what you can think about is that if you have diverse systems, I have 10 individuals in my little community here. Each one is a different species. So we can say that diversity, if we want to do the number of species, there are 10 species in my community, compared to a less diverse community where we still have 10 individuals, but they only exist in 4 different species. So we only have 4 species in that community versus 10 in our more diverse one. But we weren't just interested in species diversity, we were also interested in thinking about genetic diversity. And really, genetic diversity is this part of within species variation. So I'll start there and then try and explain what we mean by genetic diversity. And so when you think about within species, we can think about variations that are sometimes really easy to see and sometimes not so much. So easy to see would be that we have different breeds of dogs. They're all dogs, they're just different breeds. Some are small, some are big, all different kinds of traits. We can also think about how trait variation within species, so these are 2 Olympians, one is very short, one is very tall. They are both gold medalists. And this kind of within species variation can be really important in marine systems because often they're dominated by there are many coastal systems that are dominated by single species. And so in salt marshes, they're typically dominated by Spartina alterniflora. In some seagrass beds, especially up north, they're dominated by Zostera marina. So these are single species that can also very much vary in their traits, just like the dogs and just like us. And so we can think about this variation within species as being important. And so if we think that all species are beads, or beads are our species, we could have some mixtures where basically everything is a pink bead, basically everything is a orange bead, or a white bead. And so these are very low diverse communities of beads versus one where we have tons of multicolors. So it's basically the same idea as species, but we're thinking about it from within species variation. And within the group of ACER projects that we've been doing, most of this has been focused on like clonal diversity in plants. So plants grow with the rhizome, and so you can basically collect a single shoot and propagate it off of that rhizome, and over time you end up with this stock of different clones that you can look at how having many clones or only few clones, how that affects these ecosystems. The other group that did look at genetic diversity was in oysters, and so oysters are not clonal plants. And so a different way to think about this is you could have different families. You could have populations that come from one family line versus populations that would come from multiple different family lines. And so that's kind of what I mean by genetic diversity. And so we have species diversity and genetic diversity that we're looking at. Okay. So why should we focus on biodiversity? Why might this matter in the context of disturbance and how communities or ecosystems respond to this disturbance? So there's a lot of work that's been done to show that diversity can have these positive ecosystem functions, such as increased productivity, but also it can increase the stability and resistance to disturbance in communities. And so if we think about it in non-oiled, so we have low diversity and high diversity might have a little bit more of a positive ecosystem response. So this could be productivity or survival or something like that. But in the oil, what we can see is that in these low diversities, so everything is negative, oil had a negative effect, but the decrease is much greater in this low diversity from non-oiled to oiled compared to this high diversity treatment where we have a little bit less of a reduction there. So there might be some benefit to having more diverse communities in reducing this response to oil.
So you talk low diversity, we're talking about
Yep.
this one.
Yep.
That has orange beads.
Mostly
And
orange
then
beads.
When you talk about high diversity, we're talking about the one that has multicolored beads.
Yes.
So they're is more species or more genetics to look at.
Yes, and so the idea of why this might matter, or one of the easiest ways to think about why this could actually happen, is if you think that species or genotypes will vary in their response to the disturbance. So some might be more resistant or less resistant to oiling, or they might recover faster or slower. And so if we come back to our big species, say that the stingray or ray of some sort and turtle were very highly susceptible to oil. So these ones, as soon as the oil spill came in this community, they're gone. Okay, we still have 8 other species, but in this community, if these ones all die off, we're left with way lower number of species or number of individuals to begin with, and even less diverse population. And so that can be one of the aspects how diversity could actually create this pattern that I showed you here.
So let's go to Ava. She's 12. She asked, what caused the oil spill?
I don't actually remember. It was a blowout of the well, and what caused that, I don't actually know. It was an explosion of some sort and reason. I'm sorry.
The explosion damaged the well, and that's what created the oil to spill into the Gulf of Mexico.
Okay.
And so when we talk about biodiversity and ACER, how many different communities, species or genetic communities, has ACER looked at in 10 years?
Oh, here, that's my next. So we looked, there were 7 different subgroups that looked at basically ranging from microbes and phytoplankton all the way up to fish and crabs, and thinking about how communities of these different organisms influenced the response to the oil spill. And so these ACER subgroups all did field surveys. And so ACER focused on the Chandeleur Islands, which is a chain of islands off of the Louisiana coast. And these islands, they varied in their oil exposure. So some areas had high, just like that earlier map I showed you, some had high levels of exposure and some had low levels of exposure, just like that.
And so we're looking at, is it over here?
It's right there, I think. Ooh, it's hard to see. Yeah, I think that's it. And the other good thing about the Chandeleur Islands is I believe they are either a bird sanctuary or wildlife refuge of some sort, and so cleanup activities were really minimal on these islands, which means that, so a lot of the ways that I showed you how we clean up, especially I mostly work in wetlands, it can also be really destructive to the habitat. It's obviously good to get the oil out of there, but it also creates other potentially confounding factors that you don't really know if what happened was because of oil itself or if it was due to some of the cleanup activities. And so because those didn't happen, because it was a sanctuary, we chose the Chandeleur Islands to look at for all of our different subgroups. And then beyond doing field surveys, so this was essentially started in 2015, so five years, or is 2016? 5 years after the oil spill, go back out and survey, look at what is the actual biodiversity in these ecosystems now today. But we also did a series of experiments here at Dissel where we were able to manipulate diversity and oil exposure and then see how these communities responded to that exposure.
Which project did you work on?
I worked on the wetland project, which was done in all these mesocosms out here. And I'll tell you a little bit about what we did in the field survey, and then I'll come back to talking about what we actually did in the mesocosms out here. And so, so I work in wetlands, and so most of them, like I showed you earlier, that picture of a marsh, are dominated by Spartina alterniflora up here in the northern Gulf of Mexico. But with essentially increases in winter temperatures, we're seeing this expansion of mangroves, which are our tropical tree species essentially, that have been moving northward into the Gulf. And so we were really able to focus on not just what is the diversity in marshes now, but also thinking about what is in these natural stands that either are just marsh plants, Spartina, or just mangrove plants, Avicennia. What about in their mixture? So when we have these two species growing together, do we see also this reduced impact of oiling 5 years after after the spill. And so that's this. So we picked sites that had just Spartina or Spartina dominated. We picked sites that had Avicennia dominated. And then we picked sites that were a mixture of both of them. And so at all of these sites, we sampled twice a year for 2 years. And we would measure a whole bunch of plant growth measures. So we'd measure height, density, biomass, plant species richness. We also measured leaf growth and below ground biomass growth. And then at the same time, we were also interested in what are the creatures or critters that are living in these habitats. And so to do that, we did this section sampling method where you put in these bins into the sediment. You use this section sample to suck up everything that's in there. You get it back into your sieve, you take it to the lab, and you identify all the critters. So we found a lot of shrimps and amphipods and maybe some isopods, and those are the kinds of things that we did in the field, in which most of the other groups did something very similar to what we were doing.
So Rihanna, who's 6, asks, is the oil from the Deepwater Horizon spill still turning up on the beach?
I don't know about the beach, and I also I can't speak to right now because I haven't been out in the field, but the last time we were out here in the Chandeleur Islands, we did see, when we would walk through one of the oiled sites, that there was oil when you stepped in the sediment that would come up. And so I know also in Louisiana, they've re-sampled the sediment in the wetlands, and they do continually see slightly elevated levels, but I don't know all the details about that.
Poppy asks, did the oil spill create a dead zone? Do y'all know, has research looked into a dead zone in the area?
I don't know. That would be not really a wetlands thing. That's more of what's it called? Pelagic, off, essentially a little bit of not exactly the near shore habitat. So I'm not 100% sure for that. We do get dead zones in the Gulf due to the influx or removal of fresh water, but that's not really my area of expertise.
And when you talk about ACER, and let's go back and look at, so ACER looked at biodiversity.
Yep.
And ACER was one of how many consortiums that are looking at oil spill research?
Each state at least had one, a Gulf state, and there might be more than that. And there were multiple funding rounds. So there were some that had funding and then didn't have it at the next one. So over the 10 years, that's at least, was there 5, 6 Gulf states? There was a lot of people who worked on this, and a lot of people who brought in expertise for people who weren't necessarily on the Gulf as well to do some of these other aspects of a lot of this research. And really, like, what's been interesting is that now we're kind of like bringing together all that we've learned in 10 years from these different groups, and we're trying to focus on, there's bigger projects beyond just ACER, where we're taking what we learn and we're meeting up with other people in these other consortiums, and we're trying to figure out what happened to wetlands, or they're doing what happened to the near shore open ocean, and trying to bring all this together to know what do we learn with all the science that happened after the BP oil spill, and then what are the gaps that we still have to know in case this happens again, and also have these things recovered to the state that they were before. And that's a little bit hard because most of these ecosystems didn't have really good baseline data before the spill happened, but at least what will be with all this field surveys and all these experiments that everyone's been doing, if this ever happens again, we're going to have really good data on what is in the Gulf of Mexico, and then how it might change with any other type of disturbance, whatever that might be.
And so we have 2 more questions, and we'll wrap up after that. Did ACER work with the Deep End Consortium? Do you know?
I don't think so. I don't think so.
We'll have to go
Concord. I was going to say I know Concord is one of them, and yes, sorry.
And then a wetlands question: did the oil cause any mutations, genetic damage to organisms in the wetlands? From the research that y'all have.
I don't know necessarily about that. What we do see, and what some other people have seen, is that there is change in genetic diversity within Spartina itself. That's what I know most, so I'll focus on that. And it's really unclear if it's it's unclear really, because it's just survey data, if it's that things colonized after the fact and they're just different than what was in the reference marshes, or if it's some genotypes are better or more resistant to oil than not.
And that goes to the idea of before the oil spill, there wasn't this baseline data that y'all have been working to collect at this point.
Yeah, yes.
So, well, Robin, we appreciate you taking some time.
Thanks. Hopefully I taught you a little bit about what's going on with ESER at DISL and with the oil spill research that's still ongoing today.
Wonderful.
Thank you.